Welcome back, friends, as we are going to continue with our sessions of pathology, and today here we have the session 2. In today's session I want us to start discussing first about some of the basic concepts in pathology. In the first session we looked at the introduction to pathology. Now in this session we're going to start discussing with some of the basic concepts in pathology. Now this book it is the Hashimoan textbook of pathology, 6th edition. So you can just go in my Telegram group and then you can get this book for free. But for the sake of today's session, I want us to start discussing with how the cell tend to have its normal structure and then it undergo different changes due to different pathological conditions. So what we are saying here, this chapter 3 explains about the cell injury and the cellular adaptations. But first we need to know that in the body, as I said in the first session, that when we are studying pathology, we are studying just the structure of the cell and what changes they are taking place in the cell. So cell, they are basic unit of tissue. Cell, they are basic unit of tissues which form organs and systems in the human body. So we all know that organization of cells, they form the organ, and organization of cells, similar cell performing the same function, they form the tissue, and then organization of tissue, they form the organs, and then organ system, and later at the end of the day the organ system forms an organism. So cells, they are the basic unity of tissue. They are the basic unity of tissue. Traditionally, body cells are divided into two main types. The first type, which is epithelial type, and then we have the mesenchymal, epithelial and mesenchymal cell. So in health, the cell remain in according with each other. However, in 1853, Virchow first published the cellular theory of disease. Virchow, who is the father of modern pathology, he published the first theory about the cellular theory of disease, bringing in a concept that disease occur due to abnormalities at the level of cell. So these are very important for you to know that the disease occur due to abnormalities at the level of cells. Since then, study of abnormalities in structure and function of the cell in disease has remained the focus of attention in understanding of disease. Thus, most forms of disease begin with the cell injury, followed by consequent loss of cellular function. So, it is very important for you to understand this, that most of disease, they begin with cell injury. Cell injury is defined as a variation of stresses a cell encounters as the result of changes in its internal and external environment. So, in the internal and external environments of the cell, a cell can encounter different kind of factors, and those factors, they can lead to variation in its normal function. So that kind of variation is what we call a cell injury. So in general, cells of the body have inbuilt mechanism to deal with the changes in the environment to an extent. So tunaposema cell injury or cell death, we don't mean kwamba cell ikipata tu mtikisiko kidogo itakufa. No, there is an extent it can tolerate. Na ndio maana tunaenda kustudy kwamba kuna reversible cell injury and irreversible cell injury. Reversible manake you have damaged the cell but to a small extent, so it can repair. But if the cell is damaged to a higher extent, it cannot repair. So in general, cell of the body has inbuilt mechanism to deal with the changes in the environment to an extent. The cellular response to stress may vary and depend upon the following variability. So cellular response to stress depend on first the type of cell and tissue involved. the type of cell and tissue involved. It is either epithelial or mesenchymal. And second is the extent and the type of cell injury. Extent and the type of cell injury. So the type of cell which is involved and the extent and type of cell injury. So if the stress had the higher has the higher magnitude, that means it is easier for it to kill the cell as compared to when the stress had the lower magnitude. Now, various forms of cell response to cell injury may be as follows. First, when there is increased functional demand, kama cell ameongezeka functional demand, manake cell yataadapt. How does it adapt? Amehitajika afanye kazi sana, kwa either anaweza ka increase number of cell au anaweza ka hypertrophy ye mwenyewe. So the cell may adapt to changes which are expressed morphologically and then revert back to normal after the stress is removed. Kwa hizi hapa zinaitwa cell or cellular adaptations. Tutaenda kuzistudy huko mbele kwenye next session. In this session we are going to study about the cell injury. So kama cell atakuwa undergo stress due to increased function or increased demand, functional demand, that is physiological, it is not pathological. So increased functional demand, cell will undergo adaptation. But when the stress is mild to moderate, the injured cell may recover. We call it as reversible cell injury. While when the injury is persistent, cell death may occur. So if the stress extent or if the stress is severe, that means cell will die completely and irreversible cell death will occur. So the residual effect of reversible cell injury may persist in the cell as evidence of cell injury at subcellular level. That is what you call as subcellular changes. Or metabolite may accumulate within the cell, what you call is intracellular accumulation. All of these will take the independent session. We'll have the session of cell adaptation. We have the session of cell injury in this one. Also, we'll have the session about the subcellular changes, or the session about the intracellular accumulations. All of these, they are different mechanisms through which a cell can respond to stresses. Now, in order to learn the fundamentals of disease processes at the cellular levels, it is essential to have an understanding of the cause and the mechanism of cell injury and the cellular death or cellular adaptation. So what causes cellular injury and cellular adaptation? It is important for us to know the mechanisms behind, which can be best understood in context of basic knowledge of normal structure and functioning of the cell below. So first you need to know about the normal structure of the cell. Normal structure of the cell. Having known the normal structure of the cell, it will be easier for you to understand how cell death occurs or how cell injury occurs. Now you need to know about the normal structure of the cell. I know you have studied it in your first year biochemistry, and you have studied it in your advanced biology. But knowing the normal structure of the cell in pathology is very important. Now, I would like in this session, let's discuss about the normal structure of the cell first, and then in the next session we start discussing the cell death, cell injury, and then we'll continue with other topics. So, different types of cell of the body possess features which distinguish one type from another. However, most mammalian cells have a basic plan of common structure and function, except the red blood cell, which is devoid of nucleus, and its structure is described separately. So, the structure of the red blood cell is described separately because of its function, and it is devoid of nucleus. That means it doesn't have nucleus. Now, let's start discussing some of these basic cell organelles. So in cell structure, under normal condition, cells are dynamic in its structure, existing in a fluid environment. A cell is enclosed by a cell membrane that extend internally to enclose the nucleus and the various subcellular organelles suspended in cytosol. As we know that the cell membrane or the plasma membrane tend to enclose the cell, but inside there are some of the other organelles which tend also to be enclosed by the cell membrane or by the membrane which is similar to the cell membrane, which is a phospholipid bilayer in structure. Now, let's first start discussing about the cell membrane. Electron microscopy has shown that cell membrane or plasma membrane has a trilaminar structure having a total thickness of about 7.5 micrometer and is known as unity membrane. Now the three layers consist of the two electron dense layers separated by electrolucent layer. So there are two electron dense layers and they are separated by the electrolucent layer in between. Biochemically, the cell membrane is composed of the complex of the mixture of phospholipids, glycolipids, cholesterol, proteins, and carbohydrate. So these carbohydrates simply they are glycolipids because they are carbohydrate linked to phospholipids. So we have phospholipids, glycolipids, cholesterol, proteins, and carbohydrate. These layers are in gel-like arrangement and are in constant states of flux, and they are moving. That's why we call it the fluid mosaic model. The outer surface of some of the cells show a coat of mucopolysaccharide form a fused layer called glycocalyx. This is only present in some types of cells. Now, proteins and glycoproteins of the cell membrane may act as antigen, such as blood group antigen, they are proteins and glycoproteins present in the cell membrane, or may form receptors, such as in the viruses, bacteria, and bacterial products, hormones, immunoglobulins, and many enzymes. So, this is the diagram which explains what can happen to the cell due to different kinds of stresses. If we have the normal cell and then it can have some of the altered functional demand, this is physiological, then it can undergo adaptation, atrophy, hypertrophy. We discussed this. After the stress is removed, the cell will restore its normal structure and function. But we can have the mild to moderate stress. In mild to moderate stress, there will be a reversible cell injury, degeneration, subcellular alteration, and intracellular accumulations. But if the stress is removed again, the repair and healing occur. But if we have the severe and persistent stress, irreversible cell injury occur, and then this will result to cell death. Now let's continue about our discussion about the normal structure of the cell. So this is the diagram which tends to summarize the normal structure of the cell. You have the glycocalyx, you have the cilia, then you have the zona adherens. These are just structures which tend to attach one cell with another. We have microfilament within the cell, Golgi complex, phagosome, lysosome, polysome. Polysome simply means the group of ribosomes. We have the large endoplasmic reticulum with the ribosomes attached. We have the nucleus with the nucleolus within. Then we have the smooth endoplasmic reticulum. We have the free ribosomes, glycogen which tend to store food, mitochondria, and then basement membrane. Later we have the collagen. So this is the apical membrane, apical end of the cell, and this is the basal end of the cell. Now let's continue about discussing about the different organs and their function. Our receptors, they are probably related to the microtubules and microfilaments of the underlying cytoplasm. So the receptors present in the cell membrane, they are related with the other subcellular organelles which are responsible for the transport within the cytoplasm. The microtubules connect one receptor with the next. The microfilament are contractile structure so that the receptor may move within the cell membrane. Microfilament, they are contractile so that they can move the receptor through the cell membrane. Band of microfilament along with the cytoplasm and the protein of the cell membrane may form projections on the surface of the cell called the microvilli. So the microfilament, they can form elevations on the cell membrane, they call the microvilli, which are especially numerous on the surface of the absorptive and the secretory cells, such as small intestine mucosa, and increase their surface area for absorption. Now in brief, the cell membrane performs the following important function. First is selective permeability, that includes diffusion, membrane pumping, sodium pumping, and the pinocytosis, that is cell drinking. So you can see it is about selective permeability, includes diffusion, membrane pumping, and pinocytosis, that is cell drinking. Then it bears membrane antigens such as blood group antigen and the transplantation antigens. These are responsible for different functions in the cell. Then possesses cell receptor for cell recognition and communication. These are the three basic functions of the cell membrane. Now we have the nucleus, which is the mother body of the cell. Nucleus consists of an outer nuclear membrane enclosing the nuclear chromatin and the nucleoli. We are starting discussing about the nuclear membrane. The nuclear membrane is an outer envelope consisting of two layers of unit membrane which are separated by 40 to 7 nanometer wide space. The outer layer of nuclear membrane is studded with ribosomes and is continuous with the endoplasmic reticulum. The two layers of nuclear membrane at spaces are fused together, forming circular nuclear pores which are about 50 nanometer in diameter. And the function of the nuclear pores is to allow different substances which are manufactured within the nucleus to move out into the cytoplasm, because nucleus is the mother body of the cell. It is the brain of the cell. It controls all activities of the cell. Everything which is taking place in the cell is controlled by the nucleus. Now, we are saying that the nuclear membrane is crossed by several factors which regulate the gene expression and repair the DNA damage as soon as it occurs. So, different mechanisms they are done in the cell so that to make sure that it tends to regulate all activities which are taking place within the cell. And then we have the nuclear chromatin. Nuclear chromatin, the main substance of the nucleus, is composed by the nuclear chromatin. Chromatin is simply the combination of DNA with the protein histone. And after combination of DNA with the protein histone, then the DNA with the protein histone later they coil and they form chromosomes. So actually in normal cell, chromosomes they do not appear because the DNA is not coiled so that it can be very dense. But what appears in the normal cell, it is chromatin, which is in the form of the shorter pieces of thread-like structure called chromosomes, of which there are 23 pairs. In human being, we have 46 chromosomes equivalent to 23 pairs. So a nuclear chromatin tend to together measuring about 1 meter in length in human diploid cell. Of these, we have 22 pairs of 44 chromosomes which are autosomes. That means they control other characteristics of an organism. But we have one pair equivalent to 2 chromosomes which are either XX for female or XY for male. These are what we call the sex chromosomes. So we have the autosomes and we have the sex chromosome. And all of these, they are performing, autosomes, they are controlling different characteristics, inheritance of different characteristics. But the sex chromosomes, they are controlling the inheritance of sex. Now each chromosome is composed of 2 chromatids connected at the centromere to form X configuration, having variation in location of centromere. So we have the short and long arm of the chromosome, but this is not our concern. Our concern was just to discuss about the basic structure of the cell and different function of different parts of the cell. So depending upon the length of the chromosome and the centromeric location, 46 chromosomes are categorized into 7 groups from A to G according to Denver classification adopted at a meeting in Denver, USA. So we have group A to G depending on the position of the centromere and the length of the P and the Q arm. So chromosomes are composed of 3 components, each with a distinctive function. There are deoxyribonucleic acid comprising of about 20%, ribonucleic acid about 10%, and the remaining 70% consists of nuclear proteins that include a number of basic proteins, histone, neutral proteins, and acidic proteins. So we have basic proteins which are histone, then you have neutral proteins and acidic proteins, DNA, RNA, and proteins. DNA 20%, RNA 10%, and then proteins 70%. But the one which is important among all of these is the DNA. DNA of the cell is largely contained in the nucleus. The only other place in the cell that contains small amount of DNA is mitochondria, or in plants we can say it's a chloroplast. But because here we are studying about human being they don't have chloroplast. Now nuclear DNA along with histone, nuclear protein form bed-like structure called nucleosomes, which are stored along the coil of DNA. So nuclear DNA carries the genetic information that is passed via RNA into cytoplasm for manufacture of proteins of similar composition. So DNA is the very important concept among all of these which we are discussing in the nucleolus. Nuclear DNA carries the genetic information. During cell division, one half of the DNA molecule acts as the template for the manufacture of other half of enzyme by the enzyme DNA polymerase, so that the genetic characteristics are transmitted to the next progeny of the cell. That is what we call the cell replication. Now the structure of the DNA molecule was proposed by Watson and Crick, and that's why we call it the Watson and Crick DNA structure in 1953. Consists of two complementary polypeptide chain forming a double helical strand which is wound spirally around an axis composed of pentose sugar phosphoric acid chain. So the DNA is composed of nucleotides, and the nucleotide itself has three components. It has pentose, then it has it has the pentose sugar, it has the phosphoric acid, and it has the organic base. Pentose sugar, phosphoric acid, and the organic base. So the molecule is spirally twisted in a ladder-like pattern, the steps of which are composed of 4 nucleotide bases, 2 purines, which are adenine and guanine, which are denoted as A and G, and 2 pyrimidines, which are cytosine and thiamine, which are denoted as C and T in the DNA. But in the RNA, thiamine, it is replaced by uracil. So however, A pairs specifically with T, while G pairs with C. This is the specific pairing. Adenine with thiamine, guanine with cytosine. The sequence of these nucleotide base pair in the chain determines the information contained in the DNA molecule, or constitutes the genetic code. So genetic code, I hope you have studied this concept in your advanced biology and you are still remember it. In April 2003, sequencing of human genome was completed, which revealed that 23 pairs of chromosomes in the nucleus of each human cell contains approximately 3 billion base pairs, and each chromosome contains an estimate of 30,000 genes in human genome, which carry the instruction for making proteins. So here there are many information about the chromatin, heterochromatin, euchromatin, nucleolus, synthesis of ribosomal RNA by the nucleolus, and this is the structure of the DNA by Watson and Crick. Now let's go to discuss about the cytosol and some of the organelles. When we are talking of cytosol, it is cytoplasm. So cytosol, or the cytoplasm, the jelly-like ground substance in which organelles, meaning little organs. Organelles means little organs, or organelles, they are specific components of the cell or specific subdivisions of the cell, and they are specifically performing certain functions. Now in our discussion we will deal with some of the few important organelles. We deal with the cytoskeleton, mitochondria, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosome, microbodies, and peroxisomes. And when we are discussing these organelles, it is very important for you to know that what will happen in different conditions of the cell because by understanding the function of these organelles, it will be very important and it will be very easier for you to know what will happen to these organelles in different conditions. Now, in a cytoskeleton is divided into microfilament, then you have intermediate filament, and then we have the microtubules. Microfilament, intermediate, and microtubules, and they are responsible for maintaining cellular form and the movement and the correction of different materials within the cytoplasm. So by starting with the microfilaments, microfilament, they are long filamentous structure having a diameter of 60 to 80 micrometer. They are composed of contractile proteins acting in the myosin and the base materials like parts of microtubules and the ribonucleofibers. Bundles of microfilaments are especially prominent close to the plasma membrane, forming terminal web. And the microfilament, they are the one also are responsible for the formation of microville on the cell surface membrane, which is very important in the absorption along the cell surface membrane. Now, microfilament, the important concept for you to understand is that it is composed of the contractile proteins which are acting in the myosin. Contractile proteins. So microfilament, they are rich in areas of the body such as muscles, because there in the muscles contraction is high, so presence of actin and myosin filament, they are the one which are responsible in the physiology of muscle contraction. Then we have the intermediate filament. Intermediate filament are filamentous structure 10 micrometer. So you can see microfilaments, they are smallest ones. They have 6 to 18 micrometer. But the intermediate filament, they have 10 micrometer in diameter. That means their size is intermediate. And are cytoplasmic constituents of a number of cell types. They are composed of proteins. There are 5 principal types of intermediate filament. So intermediate filament, they are of different types. Cytokeratin, found in epithelial cells. Desmin, found in skeletal muscle and cardiac muscle. Then you have vimentin, found in cells of the mesenchymal origin. Then glial fibrillary acidic protein present in astrocyte and ependymal cells. Then you have the neurofilament, seen in neurons of central and peripheral nervous system. Their main function is to mechanically integrate the cell organs within the cytoplasm. And let's see about the microtubules. Microtubules, they are long and hollow. This is very important for you to understand. They are long and hollow. Among all of these cytoskeleton components, only microtube, they are the one which are hollow. Hollow, that means they have a lumen within, about 25 micrometer in diameter. So they have larger diameter, but they are hollow. They are composed of protein tubulin, and they are making up cilia and the flagella. So cilia and the flagella, which project from the surface of the cell, they are composed of microtubules. Now remember, as I said, that microtubules themselves, they are made up of tubulin. Microtubules, they are made up of tubulin, but microtubules, they are making cilia and the flagella. So microtubules, they are enclosed by plasma membrane and are active in locomotion of the cell. Also, if you ever heard about the concept of basal bodies, they're the one which are present at the base of cilia and the flagella. They are made up of microtubules also. And if you know about the concept of centrioles, I hope you have seen this in your advanced biology. Centrioles located at the mitotic spine of the cell. There are 2 other morphologically similar structures composed of microtubules. So centrioles, they act at the microtube organization center. They are made up of microtubules. Now, one thing for you to understand is that microtubules, they are responsible for the movement of different organelles within the cell and different materials within the cell. And the second thing you need to know is that the microtubules, they tend to maintain the shape of the cell. So later we'll study about the cellular changes, subcellular changes, and we'll study the subcellular changes which are taking place in the microtubules. So it is very important for you to know what will happen to the microtubules, what will happen to the microtubules during cell injury or in subcellular changes. Then let's move to mitochondria. Mitochondria, is an organelle responsible for respiration, production of ATP in the presence of oxygen. So mitochondria, they are oval structure and they are more numerous in the metabolically active cells. They are enveloped by two layers of membrane, the outer smooth and the inner folded into incomplete septa or shelf-like ridges called cristae. So mitochondria actually they are responsible for process of respiration. Chemical and structural membranes of mitochondria are similar to the cell membrane. The inner membrane in addition contains lollipop-shaped globular structures, projecting into the matrix present between the layer of membrane. So the matrix of mitochondria contains enzymes required in the Krebs cycle, by which products of carbohydrate, fat and protein metabolism are oxidized to produce energy, which is stored in the form of ATP in a lollipop-like globular structure. So the lollipop-like globular structure, they are the one which perform different reaction of Krebs cycle and other reaction of the electron transport chain. Mitochondria are not static structure but undergo changes in their configuration during energy state by alternation in the matrix and the intercostal space. The outer membrane is however less elastic. So one thing you need to understand about the mitochondria is that mitochondria also they are involved somehow in the process of apoptosis or the programmed cell death of the cell, as how we shall discuss in the next session. So because they are involved in the process of apoptosis, mitochondria they can release calcium ions, and the release of those calcium ions can lead to programmed cell death. So in the part of subcellular changes, we will study about the effect which takes place in the mitochondria. Also we have ribosomes. Ribosomes, they are just the cell machinery for the synthesis of protein. So simply you need to understand about the concept of ribosomes. Ribosomes, they are synthesized in the nucleolytes. We are making the ribosomal RNA, then it combines with proteins in the cytoplasm to make ribosomes. But simply one thing you need to understand about the ribosome, they are cellular machinery for synthesis of protein. Then you have endoplasmic reticulum. Endoplasmic reticulum, it performs different functions. Endoplasmic reticulum composed of vesicles, and sometimes the vesicles they bud from endoplasmic reticulum to the Golgi apparatus, where they are packed with enzymes which are released outside of the cell as the lysosome. Now, one among the important thing you need to understand about the endoplasmic reticulum is that it is involved in the protein synthesis because there are ribosomes attached to endoplasmic reticulum forming the larval endoplasmic reticulum. But the important concept you need to understand about endoplasmic reticulum is that it is involved in the storage of calcium. So the release of calcium present in the larval endoplasmic reticulum and some kind of subcellular changes like that will lead to cell death. We have the Golgi apparatus, which is involved in the packing of different proteins. Golgi apparatus is involved in the packing of different proteins which are transported to different parts of the body, either they used within the body or outside the body, and the synthesis of different enzymes and the hormones they involve Golgi apparatus. Also we have lysosome. Lysosome tend to store the enzymes, different enzymes, And we have different types of lysosome depending on either it contains enzyme or digestive materials. For example, we have the primary lysosome, just the storage vacuole formed from various hydrolytic enzymes. But secondary lysosome or autophagic vacuole, it is the combination of the damaged or was out cell component with the primary lysosome, so it forms the secondary lysosome. And then we have the residual body, which are ingestible materials in the lysosome or lipofuscin. For example, we have lipofuscin. We have the centrioles or centromere, which is responsible in the process of spindle movement during cell division. And yeah, they are cylindrical media of the microtubules. So other things like intracellular communication, intracellular junctions. For example, we have the occluding junction or zonular occludence and things like that. Some of the intracellular junction, they allow the ions, or some of them they allow some of the impulse to pass through them. They are not so much important for you to understand for your level, because you'll not be asked in the exam. This marks the end of our discussion about the basic structure of the cell. Later we can go back in our book and then discuss a little about the molecular interaction between cells when we'll be discussing about the concepts of inflammation. It's very important to understand about these different chemicals which are present in the cell and how. So this is just the some other concepts such as cell cycle. You can go to read them by yourself, but also we studied them in biochemistry. So cell cycle, stages of cell cycle, where the cell can stop and things like that. You can go and review them by yourself. But by this time, this marks the end of our session. In the next session, we'll be discussing about cell death. Stay tuned and don't forget to share this session to your fellow students. Thank you.